One kidney lies on each side of your vertebral column. The kidneys are the primary organs concerned with body fluid and electrolyte regulation. Unneeded fluid and ions are excreted in the urine. Also contained in the urine are metabolic waste products such as urea and uric acid, which form during the breakdown of proteins and nucleic acid. The combination of water, ions, and waste leave the kidneys as urine, which flows through the ureters and into the urinary bladder, which will store the urine until it passes out of the body through the urethra. A renal artery supplies each kidney with blood. The blood is distributed to over 1 million nephrons, the functional units of the kidney. Nephrons are found in the cortex and medulla of the kidney. Each nephron is composed of the renal corpuscle and a renal tubule. The renal corpuscle has two parts, a spherical capillary cluster called the glomerulus and a cup-like container called Bowman's capsule, which surrounds the glomerulus. The renal tubule has three contiguous sections: the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Each renal tubule empties into a connecting tubule that leads to a collecting duct. Connecting tubules from many nephrons empty into each collecting duct. Blood reaches the glomerular capillaries through the afferent arteriole. Blood then leaves the glomerulus in the efferent arteriole, which supplies a network of peritubular capillaries. From the peritubular capillaries, capillary loops, the vasa recta, extend alongside the loop of Henle. If we think again of our recycling analogy, we can compare urine formation with throwing both garbage and unused needed materials into a bin, then taking back what we want to keep. Now, before taking the trash out, we add a final few waste items. Urine is formed in 3 steps: filtration, reabsorption, and secretion, which we will discuss in order.
Let's begin with filtration.
Blood pressure within the capillaries of the glomerulus is unusually high because the efferent arteriole is narrower than the afferent arteriole. This bottleneck effect helps to push water and solutes across the capillary walls and into Bowman's capsule. In doing so, water and solutes cross a filtration membrane consisting of the fenestrated endothelial cells, the lamina densa, and the slit pores created by the podocytes that cover the glomerulus. Blood cells and large proteins are too large to pass through the filtration membrane. As it travels along the nephron, the composition of the tubular fluid changes dramatically. Reabsorption is the second step in urine formation. Most reabsorption occurs along the proximal convoluted tubule. Reabsorbed material moves into the interstitial fluid and then diffuses into the peritubular capillaries. The last step in urine formation is secretion. Additional waste products are pumped into the filtrate. The DCT and collecting systems secrete acids, toxins, and drugs into the tubular fluid. Secretion occurs along the PCT in addition to reabsorption. By the time the tubular fluid reaches the collecting duct, at least 85% of the water and sodium ions have been removed for recycling. The tubular fluid arriving at the turn of the loop is highly concentrated after its journey along the descending limb. Unlike the descending limb, most of the ascending limb is impermeable to both water and solutes. Water is not reabsorbed here, nor can solutes diffuse into the tubular fluid. Sodium ions and chloride are the most abundant ions in the tubular fluid, and large amounts of sodium and chloride are pumped out of the tubular fluid as it travels along the ascending limb.
As these ions are pumped into the peritubular fluid, the solute concentration inside the tubule steadily decreases.
As the solute concentration decreases, fewer sodium and chloride ions are pumped out of the tubular fluid. Thus, the solute pumps along the ascending limb helps to create the concentration gradient responsible for concentrating the tubular fluid in the descending limb. This is a positive feedback loop. The pumping of solutes from the tubular fluid in the ascending limb adds to the concentration gradient within the medulla, which in turn draws additional water from the tubular fluid in the descending limb. The rise in the salt concentration of the tubular fluid reaching the ascending limb further stimulates sodium and chloride transport within the ascending limb. This phenomenon is called countercurrent multiplication. Countercurrent because the tubular fluid in the ascending and descending limbs travel in opposite directions, and multiplication because the positive feedback relationship multiplies the effect on water reabsorption in the descending limb and sodium and chloride transport in the ascending limb. The countercurrent mechanism performs two services for the kidneys. First, it is an efficient way to reabsorb solutes and water before the tubular fluid reaches the distal convoluted tubules and the collecting system. Second, it establishes a solute concentration gradient that will permit the passive reabsorption of water from the fluid in the collecting system as the collecting ducts pass through the medulla.